Welcome to Biology My Passion. I am Soumya Harikrishna. We are learning the chapter Breathing and Exchange of Gases. Transport of oxygen and carbon dioxide take place by different means. When we talk about oxygen transport, majority, that means 97% of oxygen is transported in the blood using RBC. RBC is our erythrocyte or red blood cell. It has got several hemoglobin molecules in it. The oxygen and hemoglobin, they bind together to form a structure called oxyhemoglobin, and then it is transported to the tissues. Whereas we already discussed that the solubility of oxygen is very less compared to that of carbon dioxide. As a result, only 3% of oxygen is transported in dissolved form in plasma. When we talk about carbon dioxide, there are three means of transport. That is, the first and foremost, 70% majority transported in the form of bicarbonate ions. 20 to 25% will be carried by RBC using hemoglobin in the form of carbaminohemoglobin. When carbon dioxide combines with the hemoglobin, we call it as carbaminohemoglobin. Whereas only 7% will be carried in the dissolved form in plasma. But compared to this is more, 7% is carried. RBC has the pigments called the hemoglobin molecules. The hemoglobin structure, if you see, it is a quaternary structure of protein with 4 globin chains. Globin is the protein part of the hemoglobin. 2 alpha globin chains and 2 beta globin chains are there. But each globin chain has got a pigment part called heme. Heme means what? Iron. So there is an iron group in the middle of each of the hemoglobin. Each ion can bind to one molecule of oxygen. So first one molecule of hemoglobin, that is one heme. 4 hemes are there. First heme will bind with oxygen to form HbO2. Then to this HbO2, the next oxygen will bind to form HbO4. HbO4 will now bind with another oxygen to form HbO6. HbO6 will now bind with the last molecule of oxygen to form HbO8. This is the oxyhemoglobin. Now, one molecule of hemoglobin thus can carry 4 molecules of oxygen. But all the 4 are not going and binding together as I suggested. First one will bind, then the second. It is called a positive cooperativity, we say. Cooperativity means by adding one, more and more will come. For example, imagine now I am calling, suppose you are sitting in my class, I am calling one student to come and do something on the board. First everyone will be hesitated, then one student will have the courage to come forward, and first one comes, the second child also will get motivated, okay, I can also do this, second one will come, third will come, fourth will come. So all 4 are not coming together. Okay, it's not like giving a chocolate, then all will come together. This is for solving a question or drawing a diagram on the board. Then it will be, it's like a cooperativity. One adding, more and more will bind. So this is also happening one after the other. So first one oxygen will bind, by seeing that next oxygen will bind, third will bind, fourth will bind. Okay, so this association of oxygen with the hemoglobin happens in the alveolar region in the lungs, where oxygen concentration is very high and carbon dioxide concentration is less there. We know in a human being, the average amount of hemoglobin per 100 ml of blood is 15 grams. It varies in males and females, but we are taking the average. 100 ml of blood has 15 gram of hemoglobin. 1 gram of hemoglobin can bind to 1.34 ml of oxygen. 1 gram of hemoglobin can bind to 1.34 ml of oxygen. So 15 grams of hemoglobin can bind to 20 ml of oxygen. But when I am naturally doing something, this is a normal respiration, during this time, the hemoglobin will be transporting only 5 ml of oxygen to the tissues. 15 will remain in the blood, only 5 will be given to the tissues. But when I am doing strenuous exercise, I need more oxygen. So that time, 15 ml will be given to the tissues and only 5 will be remaining in the blood. So under normal conditions, only 5 ml of blood oxygen will be given to the tissues. We will try to understand the process of oxygen transport through a story. Here, alveolus is the place where oxygen is present. Now oxygen is coming to the blood. This is the place where oxygen has landed. Maybe it's the airport or our at railway station, the tissue is the house of this oxygen. So oxygen is waiting for a transport to reach the tissues. So meanwhile, who is coming there? Hemoglobin. Hemoglobin is our hero. Hemoglobin has a crush on oxygen, but oxygen has nothing like that. The oxygen has not given much attention to hemoglobin, but hemoglobin is trying to impress oxygen and telling, yes, come, I will drop you at your place. So oxygen is little bit convinced. So one molecule of oxygen is getting inside, binding, then it likes the hemoglobin, then one more comes. See here, don't think that each oxygen is different girls, no, single girl, okay. So she likes more, then third one more and more. Now she is comfortable, okay, hemoglobin is a good guy, she is also comfortable. So they have a very good bonding, oxyhemoglobin, they both are riding and coming near her house. So before reaching her house, near that, who is there? There is CO2. CO2 is our hemoglobin's X. They broke up because CO2 has a friend H plus who was reason for this breakup, because H plus also likes hemoglobin and H plus told all the wrong things about hemoglobin to carbon dioxide. Carbon dioxide believed and they broke up. So now these both will get into the blood and they see that hemoglobin and oxygen are bound. They don't like it. They start influencing the oxygen in a wrong way, telling all bad things about hemoglobin. Now oxygen, first oxygen detached, second detached, third detached, and finally oxygen is like, okay, I'm going, I'm not interested. Oxygen will get back to its house. Now carbon dioxide and H plus are in the blood. Now they have to be transported back. How? We will see. So what influenced or what helped the bonding of oxygen and hemoglobin in the alveolar region? No carbon dioxide and H plus were there to tell wrong things, right? As a result, since oxygen was high, hemoglobin could bind with the oxygen. So high PO2, carbon dioxide partial pressure was less, H plus ion concentration was less, pH was high, temperature was less, and 2,3 biphosphoglycerate or diphosphoglycerate was also less. Whereas in tissues, when it reached tissues, oxygen level is less comparatively, but CO2 and H plus are very high, and temperature is also high, and 2,3 BPG high. All these factors together contributed to the breakup of oxygen and hemoglobin. Now we will see how the carbon dioxide is transported. We learned that carbon dioxide can be transported in different ways. 7% will simply dissolve in plasma and will be transported, and 20 to 25% will be binding with the hemoglobin and form carbaminohemoglobin, because it is binding to the globin part. That's why we call it as carbaminohemoglobin. And hemoglobin also carry, because already it's broke up with the hydrogen, but some part of hemoglobin still likes carbon dioxide. So some carbon dioxide will be carried. But majority or 70% is happening in the form of bicarbonate. How is it happening? So in the tissues there is carbon dioxide. This carbon dioxide PO2 is a high here and low here, so it will diffuse into the blood. After reaching the blood, it will enter into the carbon dioxide. Some will be in the blood also, some will in the plasma. Blood means plasma. Some will enter into the RBC. In the RBC, what happens? The water molecules are available. So carbon dioxide will combine with water to form carbonic acid. It's a reversible reaction. This happens in the plasma and RBC, but in the RBC this is more, because RBC has an enzyme called a carbonic anhydrase. You know, enzymes increase the efficiency of different reactions. So in the presence of this enzyme, the reaction is very high rate and carbonic acid will form. But carbonic acid is highly unstable. As soon as it is formed, it will split to form HCO3 minus and H plus ions. So this H plus ions, this is how this H plus, who is the friend of carbon dioxide, is forming, because H plus was the reason for the breakup of carbon dioxide with the hemoglobin. Now HCO3 minus, now what happens? It will come out of this RBC to the plasma, and it will be transported through the plasma in the form of bicarbonate. But when HCO3 minus is going, here it is H plus only, then the pH is different. So to compensate that, chlorine ions will come inside, chloride ions. This is called a chloride shift or a Hamburger's effect. Now what is left here? H plus ions are left after formation of this. So these H plus ions will bind with the hemoglobin, releasing the oxygen. So oxygen is coming to the tissues, and who will take up the place? So their H plus ion concentration and carbon dioxide concentration is actually dissociating oxygen from hemoglobin, and H plus will bind with the hemoglobin. Okay, so this is what is happening at the tissue side. I repeat, first carbon dioxide will diffuse into the plasma, then it will enter into the RBC, where it will combine with the water in presence of an enzyme called carbonic anhydrase to become an unstable acid called carbonic acid, which dissociate to form HCO3 minus and H plus. HCO3 minus will go to the plasma and it will be carried to the lungs or alveolar region. H plus will bind with the hemoglobin and that also will be carried. Now on reaching the lungs, what happens? Their PO2 is high. In the presence of PO2, this PCO2 and H plus, they will dissociate from the hemoglobin. They understood that oxygen knows everything, their plan will not work out, so they dissociate from hemoglobin. So influence of oxygen is there in the alveoli, which will dissociate carbon dioxide and H plus, whereas at the tissues, the influence of carbon dioxide and hydrogen ion will dissociate the oxygen from hemoglobin. When the carbon dioxide enters into the hemoglobin, it will dissociate oxygen and it will help in the unloading of oxygen from the hemoglobin. This process is called the Bohr's effect. Every 100 ml of deoxygenated blood delivers 4 ml of carbon dioxide to the lungs. Now we will learn about oxygen dissociation curve. It's a graphical representation of depicting percentage saturation of hemoglobin with oxygen against partial pressure of oxygen. So in the y-axis we are representing how much saturation of oxygen with the hemoglobin is happening, whereas on the x-axis we are taking partial pressure of oxygen. So we know that initially the binding is very slow. As I told, it will slowly bind because positive cooperativity, as I told, one after the other only it will be binding. So saturation is going at a very slow rate, then it will go very high and then complete saturation happens. So this is the maximum PO2. I am not taking the value now, just say whatever be the maximum, it is here. So at this hemoglobin is bound, maximum hemoglobin, 100% saturation is almost happening. So this is happening in the lungs. And you know that in the lungs, maximum oxygen combines with the hemoglobin to form oxyhemoglobin. Whereas somewhat here if you take, this is the tissue level. Here PO2 is not that very high because in tissues PO2 is less, and this is the percentage saturation of hemoglobin. That means this much oxygen, this distance, this much oxygen dissociated from hemoglobin and delivered to tissue. So this is the oxygen delivered to tissue. Okay. So if you want to deliver more oxygen, maybe till here, we have to reduce the oxygen level. But that is not good for us because we cannot reduce our oxygen level because oxygen is required in the tissue. So by reducing the oxygen, delivering to the tissue is not possible. So here we have another condition that's called the Bohr's effect, we say. That is already, as I discussed, when carbon dioxide partial pressure is high in the PCO2, is high in the tissues, and H plus is high in the tissues, that will unload the hemoglobin and oxygen naturally. So in such case, our graph is shifting to the right. That means it takes more time to get saturated, and top level will be high. Finally saturation of it will be happening, but at this level if you see, at this PO2, initially this was the level oxygen delivered, but now if you see, you have this much. So more oxygen delivered in the presence of high PCO2, high H plus, high temperature but low PO2. This is Bohr's effect. Okay, but now you know that in the lungs the oxygen is more, carbon dioxide H plus are less. As a result, the binding of oxygen will be very high. So as a result, this is the right shift. The graph is shifting to the right due to Bohr's, this is Bohr's effect. Okay. Now I am seeing that the graph is shifting to the left. That is, in this case, if it is shifting to the left, you know that more oxygen, that is at this level itself, it is reaching the maximum. More oxygen, saturation will happen easily. So this is called a left shift. It is because of high PO2, less PCO2, less H plus and less temperature. So here hemoglobin is not binding with the carbon dioxide or H plus, or affinity of hemoglobin with the carbon dioxide and H plus is less due to the presence of oxygen. That condition or that process is called the Haldane's effect. Okay, so this is called a oxygen dissociation curve. Hope you understood the concepts. If you find it useful, please like, share and subscribe to my channel Biology My Passion. Thank you for watching.